Okay, good evening, guys. We'll start the session. I'll share my screen, and one of you can unmute and let me know if I'm audible and if my screen is visible. Just give me a minute. Okay, so I'm sharing my screen. Oh no, sorry, not this one. This one. Can one of you unmute and let me know if my screen is visible and if I'm audible? Is my screen visible? Am I audible? Okay. Hello. Am I audible? I'm assuming I am audible. You can type in the chat box. I am audible, right, guys? Hello. Are you saying something I can't hear? Just type. Okay, thanks. Okay, so I'm assuming. Okay, thanks. So I'm assuming my screen is also visible. So we'll start today's session. So good evening, guys. Today is the 11th week live session of this course, Fundamentals of Protein Chemistry. So what we do in these live sessions, we take some old assignment questions and we basically dissect the questions to arrive at the answers. We not only discuss the answers, we discuss the concept behind those questions so that it's easier for us to solve similar type of other questions, right? Because that is our whole motive of doing these question answer sessions. Not only learning the answers of that particular question, but also learning the concept behind those questions so that we can solve other questions as well. That's what we have been doing in our live sessions, and that's what we'll do today also. So today's 11th live session of this course, Fundamentals of Protein Chemistry, taken by course instructor Professor Shagota from IIT Kharagpur. And so we'll try to keep the sessions as interactive as possible. So I give a question, I'll wait for you to come up with the answer with proper details. Then we'll discuss the concept and all everything. I'll wait for you to ask me doubts and we'll move on to the next question. So we want these sessions to be as interactive as possible. So if you have any questions, you can either unmute yourself, let me know, or write in the chat box. If I know the answer at the moment, I'll definitely tell you. If I don't, then you have to give me some time. I have to do some reading and then I'll let you know the answer by the next class for sure. Okay, so with that note, let's start with the first question for today. So the first question for today is, which of the following techniques is best suited for separating proteins based on their size? Okay, so I'll repeat the question again. You can already see it on your screen. Which of the following techniques is best suited for separating proteins based on their size? A. Ion exchange chromatography. B. Affinity chromatography. C. Size exclusion chromatography. And D. Thin layer chromatography. So you have to answer, give a detailed answer for this, okay? Not only the option C. Anybody else? Correct. Any other names for size exclusion? Do you know how what size exclusion is called uh, differently? Like correct. SEC or size exclusion chromatography. So basically, the correct answer here will be SEC or size exclusion chromatography, which is also known as gel permeation, gel filtration chromatography, because it separates proteins based on their molecular size or hydrodynamic volume, right? So large molecules elute first because they cannot enter the pores of stationary phase beads, and smaller molecules enter the pores and therefore elute later. But why not the other options? So A, ion exchange chromatography basically separates uh, substances based on their charge, not on their size. You can see ion exchange chromatography. So ions are involved. So basically charge is involved, not really size. For affinity chromatography, it separates based on specific binding interactions between a protein and a ligand. Affinity for a very particular affinity is being exploited in case of affinity chromatography. And for thin layer chromatography, basically it's used for small molecules and separates based on polarity, not really suitable for large proteins. Okay, so therefore size exclusion chromatography is the best choice for separating proteins based on their size. So if we go a little bit about gel filtration chromatography, it's basically a separation technique which is strictly based on molecular size. It's helpful method to separate the macromolecules of different sizes from one another. It's also known as gel permeation, gel filtration, size exclusion, molecular size exclusion, or molecular sieve chromatography. So water is a mobile phase in gel filtration chromatography, whereas organic solvents used as a mobile phase in case of gel permeation chromatography. So it's basically nothing but a special case of partition chromatography, because the stationary phase is the same as the mobile phase. Stationary phase in the form of porous, when a sample containing solutes varying from small to large molecules leads to the column. Small molecules penetrate all the pores and are retained, thus being eluted later than large molecules which move only in mobile phase. Molecules of intermediate size penetrate only some pores, thereby being retained to a lesser degree than smaller one. This will be lesser, not lesser anyway. The elution of the solute is best characterized by a distribution coefficient which is called Kd. One second. I don't know what is this type. Okay. So Kd is a distribution coefficient. One second, give. Yeah. So Kd will be the volume of solvent required to elute an unretained compound, also called as the void volume. Not Kd. Kd is a distribution coefficient VO. VO will be the volume of void volume. VE will be the volume of mobile phase required to elute and compound rather than from the chromatographic column. Okay, so the best known media for gel filtration are based on dextran, a polymer of glucose cross-linked with epichlorohydrin. So these are called Cephadex. So degree of swelling determined by the degree of cross-linking. Others can be agarose bases as Cepharose, mixed type, Cephar or agarose and polyacrylamide like Cephacryl. So in case of Cephadex, Cephadex types are designated by G, followed by a number which corresponds to 10 times the water regains. So the number of millimeters of water taken by 1 gram of dry gel matrix. Right? So that's the commercially available series will be Cephadex G10, G15, G25, G50, G100, G200. Their fractionation ranges for proteins with molecular weights up to 600,000 daltons. So it also swells in organic solvents such as dimethyl sulfoxide, formamide, dimethyl formamide, and mixtures of lower alcohols, but the degree of swelling in organic solvents. Now, Cephadex also swells in organic solvents such as dimethyl sulfoxide, DMSO-formamide, dimethyl formamide, and mixtures of lower alcohols with water. However, the degree of swelling in these organic solvents or their mixtures is less than in water alone. The Cephadex series LH20, LH60, and Cephacryl are suitable for organic solvents. Cephadex LH types are specifically prepared for gel filtration in organic solvents. So these are prepared by the hydroxypropagation of Cephadex G50, and these both have hydrophilic and lipophilic properties. In case of Cepharose, this is based on agarose. It is usually used mostly for the fractionation of large molecules. The agarose base is obtained by the purification of agar, that is devoid of charged polysaccharides, and the polysaccharide chains in agarose consist of D-galactose and 3,6-anhydro L-galactose, forming double helices like DNA which aggregate on aging to form bundle-like structures. So these bundles are mechanically strong with pore size capable of fractionating macromolecules of molecular weight approx. Okay, so Cefacryl is an example of mixed gel, agarose and polyacrylamide, and the agarose forms a support frame for the polyacrylamide gel. So this is basically the size here. Up to 700 is for G10, up to 1500 daltons is for G15 and all of that. So accordingly, depending on the separation limit you want to exploit, you can take which type of Cephadex you want. Similarly, now what should be the characteristics of the gel matrix? Very important. The characteristics of gels are enumerated below, like chemically inert. The gel matrix should be chemically inert, and any chemical interaction between the matrix and the cellulose may lead to irreversible binding to the gel material and chemical alterations of the substances. The matrix must be stable over a wide range of pH and temperature and control particle size and size distribution. So the particle size must possess good flow rate characteristics. Therefore, it is essential that the particle size distribution be as narrow as possible. Right, and should be mechanically rigid. This is essential for the gel particles so as to prevent deformation by the forces caused by the flow of liquid. So there are many different applications of gel permeation chromatography. So these have been employed in biomedical research problems and clinical laboratories. So these include separation of polysaccharides, enzymes, antibodies, and other proteins. Separation of non-polar species such as triglycerides in non-aqueous mobile phases. Then separation of non-polar species such as the desalting process like removal of salt and buffer from protein preparations as alternative to dialysis, phenol removal from nucleic acid preparations, then removal of products like cofactors and inhibitors from enzymes and all of that. So chromatography is a technique for separating mixtures into their components in order to analyze, identify, purify or quantify the mixture components. Okay, so this is a mixture, you separate the components, then you can take them forward for analysis, identification, purification, quantification, whatever. So chromatography is used for analysis to examine a mixture, its components and their relations to one another. It is also used for identification, determine the identity of a mixture or component based on the other known components. Then purify, separate components in order to isolate one of interest for further study. Then quantify, like determine the amount of the mixture in or the components present in the mixture. This is basically quantification, nothing else. Now, real life examples of uses for chromatography. Pharmaceutical, like determining amount of each chemical found in new product. In hospitals, people detect blood or alcohol levels in a patient's bloodstream to compare sample formed in a crime scene that is used in forensics and law enforcement and all of that. So detailed definition will be, it's a laboratory technique that separates components within a mixture by using the differential affinities of the components for a mobile medium and for a stationary absorbing medium through which they pass. Differential means showing a difference, a distinctive, right? Affinity will be natural attraction or force between things. Like mobile medium, gas or liquid that carries the component, that is called the mobile phase. The moving phase. Stationary is the phase that is constant, not constant, this phase that is not moving basically, that does not move with the samples. It's a non-mobile phase of the stationary phase. Now, the explanation compound is placed on the stationary phase. Mobile phase is passed to the stationary phase. Mobile phase will have to solubilize the compounds, the components, and mobile phase carries the individual components certain distance to the stationary phase depending on their attraction to both the phases. So this is a mixture. This is how separation will happen. A very simple schematic of illustration of chromatography. So you see over here, this is thin layer chromatography basically. So depending on the solubility and polarity and all of that. So there can be liquid chromatography, separates liquid samples with the liquid solvent, like mobile phase will also be liquid, and a column composed of solid piece like stationary phase. Gas chromatography separate vaporized samples with a carrier gas and a column composed of a liquid or of solid beads. That is GC. Okay. And then paper chromatography separates dried liquid samples with a liquid solvent and a paper strip, which will be the stationary phase. Then we have the thin layer chromatography that separates dried liquid samples with a liquid solvent and a glass plate covered with a thin layer of alumina or silica gel. Now, what is capillary action? The movement of liquid within the spaces of porous material due to the forces of adhesion, cohesion, and surface tension. So the liquid is able to move up the filter paper because its attraction to itself is stronger than the force of gravity. Right? Then we have solubility, the degree to which a material or solute dissolves in a solvent, and solutes dissolve into solvents that have similar properties, like dissolve like. This allows different solutes to be separated by different combinations of solvents. Separation of components depends on their solubility in the mobile phase and their differential affinity to the mobile phase and the stationary phase. So these are the different types of chromatography. We have the gel filtration or the size exclusion chromatography, which separates based on size. Hydrophobic interaction chromatography, ion exchange chromatography is based on charge. Affinity, specific affinity between two compounds like nickel and histidine. That's how the nickel and T affinity chromatography is performed. Then we have the reverse phase and all of that. So, if you talk about the affinity chromatography, there's an historical aspect as well, where this particular pharmacologist, Emil Starkenstein, published a paper on the influence of chloride on the enzymatic activity of liver amylase, and generally considered to be responsible for the first experimental demonstration of the biospecific absorption of an enzyme onto solid substrate. But not long after, Will Statter also enriched lipase by selective absorption onto powdered stearic acid. So it is known as today affinity chromatography. So it offers high selectivity, hence high resolution and usually high capacity for the proteins of interest. Purification that would otherwise be time consuming, laborious, difficult, or even impossible using other techniques can be easily achieved by affinity chromatography. So, technique can be used to separate active biomolecules from denatured or functionally different forms to isolate pure substances present at low concentrations in large volumes of crude samples, and also to remove specific contaminants. So first is the induction of the mixture, second is the absorption of the second is absorption of the object materials, third is washing out of the impurities, and fourth is elution of the object materials. This is how basically it works. So affinity chromatography separates proteins on the basis of reversible interaction between a protein and a specific ligand coupled to a chromatography matrix. Okay. So like this. So purification steps, equilibration, buffering, and all of that. The first is so basically the steps are binding. Buffer conditions are optimized to ensure that the target molecules interact effectively with the ligand and are retained by the affinity medium as well as other molecules washed through the column. Elution is buffer conditions are changed to reverse or weaken the interaction between the target molecules and the ligand so that the target molecules can be eluted, separated from the column. Washed before that, buffer condition that wash unbound substrates from the column window without eluting the target molecules, or that is re-equilibrates the column back to the starting conditions. In most cases, the binding buffer is used as the wash buffer. Ligand coupling, covalent attachment of a ligand to a suitable pre-activated matrix to create an affinity medium. Then pre-activated matrices basically which have been chemically modified to facilitate the coupling of specific types of ligand. Okay, there can be different kinds of matrix, ligand immobilizations, spliceogram. So, what are matrix? Proper selection of a matrix or carrier for ligands is of decisive importance for the successful application of the specific absorption. Obviously, like insolubility, sufficient permeability, high rigidity, suitable particle form, zero absorption capacity, chemical stability, and other conditions required for absorption. All of these are necessary things to look out for. Resistance towards microbial and enzymatic attack, good flow properties for rapid separation, all of that. So no matrix is supported ideally in all aspects. Porous, glass, cellulose, polyacrylamide, agar, some of these are matrices. So the selection of the ligand for affinity chromatography is influenced by two factors. The ligand must exhibit specific and reversible binding affinity for the target substance. It should be reversible as well because we need to elute that substance, right? And it must have chemically modifiable groups that allow it to be attached to the matrix without destroying the binding activity. Absolutely. We don't need the binding activity destroyed. So we'll not go into too much. There can be cyanogen bromide coupling, bios-oxidant coupling. We'll not go into these details. Or like a design. Next, column chromatography. Column chromatography is a technique in which the substances to be separated are introduced onto the top of a column, packed with an adsorbent, passed through the column at different rates that depend on the affinity of each substance from the adsorbent, and for the solvent or solvent mixture, and the usually are collected in solution as they pass from the column at different times. It is a solid liquid technique in which the stationary phase is solid and the mobile phase is the liquid or gas. So it was developed by American chemist D. Day in 1900, while M.S. Trent, the Polish botanist, also used adsorption columns in the investigation of plant pigments. So in column chromatography, the stationary phase is packed in a gel or metal column, glass or metal column, and the mixture of the analytes is then applied, and the mobile phase, commonly referring to as the eluent, is passed through the column either by use of a pumping system or applied gas pressure. So the stationary phase is either coated into discrete small particles like the matrix and packed into the column, or applied as a thin film to the inside wall of the column. So as the eluent flows through the column, the analytes separate on the basis of their distribution coefficients and emerge individually in the eluent as it leaves the column. So it's one of the most useful methods for separation and purification of both solids and liquids. Its major application is like separation of mixture of compounds, removal of impurities for purification process, isolation of active components, isolation of metabolites from the biological fluids, estimation of drugs, and the formulation of crude extracts. I'll go into detail. So basically, chromatography is a physical method of separation in which the components to be separated are distributed between the two phases, one of which is stationary, one of which is mobile. It moves to the definite direction. The chromatography process occurs due to the differences in the distribution constant of the individual sample compounds. So, is there any technique to separate and identify the whole components of a mixture? Works by allowing the molecules present in the mixture to distribute themselves between the stationary and the mobile medium. Molecules that spend most of their time in their mobile phase are known to have carried along faster. So classification according to their mobile phase It can either be liquid chromatography, mobile phase is a liquid, or gas chromatography. Mobile phase is a gas. It's self-explanatory. So classification according to the packing of stationary phase. So thin layer chromatography, stationary phase is a very thin layer supported in a glass, plastic, or aluminum plates. In paper chromatography, the stationary phase is basically a thin film of liquid supported on an inert support. In column chromatography, stationary phase is packed in a glass column. So classifications according to the force of separation can be adsorption chromatography, partition chromatography, ion exchange chromatography, gel filtration chromatography, and affinity chromatography. If we talk about thin layer chromatography, it's a method of identifying substances and testing the purity of the compounds. Now, TLC is a useful technique because it is relatively quick and requires small quarantine of material. Separation of in TLC involves distributing a mixture of two or more substances between a stationary phase and a mobile phase. Stationary phase is a thin layer of absorbent, usually the silica gel or alumina, coated on a plate, right? For the mobile phase, developing liquid which travels up the stationary phase carrying the samples with it, and components of the samples will separate on the stationary phase according to their how much they absorb on the stationary phase versus how much they absorb in the mobile phase. This is how thin layer chromatography is done in a beaker. TLC plate is kept and it is spotted, and then the compound moving upwards is noticed, observed. So these are some TLC images. I'm not going into too much detail. So you can go into details if you want. I'll share these slides. Okay. I talked about this. We'll come back to this if you have questions again. In case of learning any other type of chromatography. For this we'll wrap it up because we've already discussed about saturation chromatography. Okay. So sorry, I'll share these slides. If you're interested, you can go back to the slides and see what all you is. Now, why fractionation? Fractionation makes it possible to isolate groups of proteins or fractions from total proteome or proteins or fractions based from total proteome. It simplifies analysis of complex protein mixtures. It's proteins less crowded than 2D maps and allows for improved resolution when an individual fraction is analyzed. Wait, my slides are. You would have to go back to the question that we started with. Wait 1 second. One second. Just finish this question. I just. So what we started with, the right answer will be cell distribution chromatography, right? So we'll come back to the chromatography slides I have loaded here in case we need it for the other questions, or else I'll upload with the slides. I'll wait for 2-3 minutes if any of you have any questions, or we can move on to the next question for today. I'll wait for 2-3 minutes. Yes? Any question? Yeah, sure. Ask. What did you say? What inhibitor? Protease. Okay, okay, okay. PMSF, yes. So, what is your question? What is the role of PMSF? So PMSM is basically phenyl methyl sulfonyl fluoride, which is a chemical compound used to reversibly inhibit serine proteases. So it works by forming a covalent bond with the serine residue in the enzyme's active site, thereby prevents the breakdown of proteins during cell lysis. So that's why it's a common supplement that's added to the lysis buffers during protein extraction and purification buffers in research, and is often dissolved in organic solvent like ethanol or isopropanol before use. So it targets the serine proteases, a class of enzymes that will breaks the peptide bonds in proteins. So it irreversibly binds to the active site of the serine residue through a process called sulfonylation, and it forms a stable covalent bond. So this binding alters the enzyme structure, rendering it inactive and preventing it from degrading proteins. Okay. EG. EG, I am not sure. Where was it? Maybe there was a. Yes. Can you use it for? Your voice is cracking. Can you type once? If you can type, I can hear your. It's cracking. Acha, can you repeat once? Can you repeat? Yes, yes. So desalting is nothing but it's used to remove small molecules like salts from a larger molecule of interest, such as a protein or nucleic acid. So if you think about the theory like that, it's just removing small molecules like salts. So it works by passing a sample through a column packed with porous beads. Large molecules will be too big to enter the pores. They will elute quickly, while small molecules like salts will enter the pores, slow down their movement, and will be separated from the larger molecules. So the resulting column is selected with the pore size designed to exclude the large molecules but retain the small molecules. So the column is then equilibrated with the buffer or water solution. So the sample containing both the large molecule and the small molecules will be loaded onto the top of the column, and as the sample will move through the column, the large molecules which cannot enter the pores of the resin will pass through quickly, and the small salt molecules on the other hand will enter the pores, slowing down their progress through the column, and the fractions will be corrected as they exit the column. So the first fractions will contain the large molecules that were excluded from the pores and all of that. So like basically the theory stands, the small molecules will be separated from the large molecules, And for the equilibration, if you talk, equilibration will happen with organic solvents. For those kinds of organic solvents, for desalting we use acetonitrile and some percentage. I don't know the exact percentage, but in case of protein desalting, which we often use C18 columns and all, so basically the resin will be pre-equilibrated in mostly water, I guess. So basically to separate the soluble molecules from smaller molecules. Yeah. Yes, yes. That's sorry. What manufacturing? Resins, yeah, Cephodic Cephorose, yes. Am I doing what? I'm not doing in kind of resin manufacturing. No. No, no. We are not doing resin manufacturing. Thank you. Yeah. So sorry. Yeah. Okay, I'll wait for 2 more minutes. If anybody else has any questions, you can ask. Then we move on to the next question for. Okay, see if there are no questions with this, no queries on this question, we'll move on to the next question for today. The correct order, sorry, uh, the correct order of event in the general strategy for protein purification from tissue of microorganism is number 1, crude fractionation, number 2, disruption, number 3, selective fractionation. And the options are A in the order of 1, 2, 3, B in the order of 2, 1, 3, C in the order of 1, 3, 2, D in the order of 2, 3, 1. Which one? Sorry. B. 213. Okay. Anybody else? Yes, so the general strategy for protein purification from tissues and microorganisms will follow this sequence. Basically, it will be disruption, which the first step is to break open the cells or the tissues to release the proteins, right? Like any kind of sonication, homogenization, detergent lysis, etc. All of these processes that can be optimized for different cells, different tissues, the purpose of downstream application, all of that. Second step will be crude fractionation, which will be an initial separation of major class of molecules. For example, centrifugation to remove debris or ammonium sulfate precipitation, all of that. The last step, the third step will be selected fractionation, which will be finally use specific purification steps to isolate the protein of interest. It can be either ion exchange chromatography, affinity chromatography, or high extrusion chromatography. So the correct sequence of events will be disruption, crude fractionation, and then selected fractionation. So what happens basically? So in case of purifying, if you want to monitor the purification quantity, we have to quantify the total protein, and if it's an enzyme, we have to check the enzymatic activity as well. Specific activity of the enzyme, enrichment, yield, all of these can be calculated using these equations. So a successful purification step should not only increase the specific activity of the enzyme substantially, but the enzyme should also be recovered in good yield. So the yield of enzyme after a particular purification procedure may be low, not because of the procedure is failing to purify the protein, but because it is causing some inactivation of the enzyme. These are some practical tips. An additional problem sometimes experienced is that the enzyme may have been active immediately subsequent to the purification step but may lose substantial activity if stored for long period prior to assay. This happens for enzymes. So, how do we assess the purity? Pure proof that the protein is pure can only really be obtained by protein sequencing, to be very honest. However, considerably simpler analytical procedures can be used. Day to day lab procedures can also be used to provide a good indication of the degree of purity, like SDSPH, analytical isoelectric focusing, two-dimensional gel electrophoresis, all of that. So the detection method used to stain the gels is also important. For example, Kumasi blue staining is much less sensitive than silver staining, and it is possible to have what looks like a pure protein on Kumasi blue that will not look so good when the same gel is silver stained. Okay, so what is the hypothetical purification scheme? First will be the growth or the induction. Obviously, the growing of the cells, how you grow the cells, under what condition, if you induce the cells with IPTG or anything like that. Basically, to induce the production of the particular protein that you want inside the cells. Then you have to harvest the cells. You centrifuge the cells, you separate the cells from the media and all of that. Then is the lysis of the cells by sonication and centrifugation to remove the debris and precipitation of the nucleic acids. First, you need to separate the nucleic acids from the proteins if you want to isolate and purify the proteins. Then will be the ammonium sulfate fraction, dialysis, ion exchange chromatography, and concentration, gel filtration chromatography, hydrophobic chromatography, dialysis, concentration, all of that. That is what basically the purification scheme follows. So direct methods for the determination of protein concentration will be these methods, like which, but they require very pure protein and composition for like absorbance 280, which will be quite accurate. Basically using a UV spectrophotometer. Indirect method, which is dye binding, they are required for the standard, basically Bradford assay, BCA, Lowry. In this case, we use complex proteins from complex cell lysis, not necessarily any particular protein or anything like that. From complex cell lysis or complex tissues, when we isolate the proteins, we can measure the concentration using these methods. Okay. Now, if you want to overexpress any particular protein or anything, basically you have to play with the like, for example, overexpressing a particular protein can lead to the formation of the protein by inclusion bodies. That is very difficult to purify. Only a small fraction of the protein will remain soluble, and this was not fully active also. So how do we attempt to refold the protein? Like, solution will be co-expression with a soluble fragment of the chemoreceptor. So the co-expression of a substrate increases solubility. This led to the development of an efficient purification scheme. So any kind of troubleshooting you do while purification, you'll have to find out a solution for that. Like, for, as I said, for protocol, grow induce harvest cells, few proteins precipitate for this ammonium sulfate precipitation. Again, all of that wash intensively and then elution. SDS basically to purify the proteins, cell-free lysate. Lysate will obviously have a lot of proteins. If you see over here, it's not purified. The first one. This is SDS page, where you basically separate proteins according to their size as well on a gel matrix used made with acrylamide and polyacrylamide. So here also, if you see, the first one is cell-free lysate. Its cell has been lysed and the lysate is been loaded. So if you see, there are lots of proteins in the cell. That's why there are bands everywhere. But with each step of purification, each step the lysate basically gets more and more purified until you are remaining with one protein of interest that you wanted. Okay, so we'll not go into. So basically, cellular fractionation is a process you separate the cellular components and preserving their individual functions of each component. It's an important step for studying specific intercellular structure, organelle, proteins, or assess possible associations between these macromolecular structures, which provide various information like bovine density, surface charge density, then size and shape. And this is a method that was originally used to demonstrate the cellular location of various biochemical processes. So other uses of subcellular fractionation is to provide an enriched source of a protein for further purification and to facilitate the diagnosis of various disease states. It is a method that dissects cells into various organelles according to their shape, size, density, the viscosity of medium, and rotor speed under the influence of any external applied centrifugal force. Right? So, if we talk, what is the principle of centrifugation? So, the principle of centrifugation technique is to basically separate the particles suspended in a liquid media under the influence of a centrifugal field, which is the process involved. This is the principle of sedimentation. Sedimentation is the phenomenon where the suspended material settles out of the fields by gravity. Right? So basically, tissue samples which we work in labs, tissue samples or cellular samples. They are typically homogenized in a buffer solution that is isotonic to stop osmotic damage. We don't want osmotic damage. So mechanisms for homogenization, they can include grinding, mincing, chopping, pressure changes, osmotic shock, freeze thawing, all of that, ultrasonication. The samples are then kept cold to prevent enzymatic damage. The samples are kept cold so that proteins, which are also protein structures, are maintained, because protein can get denatured at high temperatures, and also the enzymatic activity is maintained, right? So it is the formation of homogeneous mass of cells like cell homogenate or cell suspension. It involves grinding of cells in a suitable medium in the presence of certain enzymes with the correct pH, ionic composition, temperature. For example, pectinase, which digests middle lamella among plant cells. So in case of plant cells homogenization, this will be used. Pectinase, this enzyme will be used. So cell homogeneous is separated into fractions by spinning them super fast in a process called centrifugation, right? So basically it produces a force that are thousands of times higher than the gravity, and the cellular components are pushed towards the bottom of the container. Okay, so centrifugation applies enough force to cell homogenates to allow different cellular fractions to separate based on the properties such as mass, density, and shape. So as shown in, there is a figure here anyway. Acceleration causes components that are too heavy to resist the force of gravity to move to the bottom of the tube, right? So if you see, this is a schematic. I'll show you better. So basically purification is achieved by differential centrifugation. The sequential increase in gravitational force results in the sequential separation of organelles based on their density. So because of the original different fractions that will be collected by centrifuging the sample several times. So every time you centrifuge, basically a separating based on the density, basically. This is how it happens. So enzyme that is known to be localizing exclusively in the target organelle, isolation of any organelle requires a reliable test for the presence of that organelle. So for example, I need to isolate an enzyme that I know is present in, for example, let's say nucleus. For example, just hypothetically speaking, an organelle like maybe ER also, or nucleus, or maybe mitochondria, right? So now if you're separating the organelle using centrifugation, and be it however, like centrifugation we have isolated after using the centrifugal force for a lot of times, several times. Now we are saying that this fraction is the mitochondria, but we need to prove that this fraction that we have in our hand is a mitochondria. For that we need some markers. So what are markers? Markers that are basically will be a markers, the presence of which will be a very reliable test that whatever you have in your hand is mitochondria. So marker enzymes also provide information on the biochemical purity of the fractionated organelles. So the presence of unwanted marker enzymes activity in the preparation indicate the level of contamination. For example, if I say that I am having mitochondria, but I get the readout of a ER enzyme, for example, from my sample, then I can not say that it is pure mitochondria. It has some contamination of the ER component as well, right? That is what, okay. So these are enzymes that are basically used as markers in the subcellular fractionation. If we are trying to separate nuclei, we will look for DNA polymerase, RNA polymerase, For plasma membrane, maybe leucine aminopeptidase, 5-nucleotidase. For endoplasmic reticulum or cytosol, glucose 6-phosphodiesterase, aldolase, cytosol aldolase. For mitochondria, basically cytochrome oxidase, succinate dehydrogenase. For lysosome, acid phosphatase, aride sulfatase C. Peroxisose, it will be catalase, peroxidase. For Golgi, what is it? Saline transferase. So there are some of the enzymes which are very prominently present in these particular organelles, and they act as markers during these in these particular organelles. Okay. Now, what is the if you talk about examples of fusion systems for protein purification? If you have done protein purification in labs, you know about the histidine, hexahistidine sequence basically. So why is it such a hugely popular thing? So based on nickel chelation by polyhistidine, several vectors and fusion times available like this. Versatile systems when accessory. So if I am trying to purify a protein, I will the gene encoding the protein, I will basically fuse 6 histidine residues to that. So when the protein is expressed at one end, it will have 6 histidine residues. So one of the steps of protein purification that we can use will be nickel NTA affinity chromatography. So nickel will have a special chelation of the polyhistidine. So based on presence of nickel will pull the polyhistidine segments towards it. Since my protein of interest is also attached or fused to this polyhistidine segment, my protein of interest will also be pulled. So similarly, the maltose binding protein fusions are there, GST fusions, flat TAC fusions are there. So these systems generate large protein fusions and are purified based on the properties of the fused proteins. So all work well, but the addition of the fusion can be a problem sometimes. Like why it can be problem with the any kind of fusion addition might create a problem in the proper folding of the protein, or it will basically lead to a bigger protein. So bigger proteins might have difficulty in getting purified and all of that. There can be some problems, but those have to be basically troubleshooting needs to be done during the purification process. Okay, so we will not go over express proteins, we will not go into. So we also use some reducing reagents because the bacterial cytoplasm is reducing, so there are no disulfide bonds in cytoplasmic proteins. Therefore, if you have cysteine residues in your protein, you should include reducing agents. Some level of reducing agents should be added for your periplasmic proteins as well, right? So, if you have no cysteine residues in the protein, then this is not so important. So, DDT and beta marcaplets are also reducing. So, as you said, proteases. So, why do we need proteases? Proteases are basically acting against you during a purification. Always follow the general rule. So proteins basically are proteins, enzymes that degrade the proteins. So if you are doing protein purification and there are proteases in your sample, it will basically degrade the protein you are trying to purify. Okay, so what we need to work fast, especially during the early steps until most of the proteases are separated from the protein. Many of the cells most active non-specific proteases are in the periplasm, and problems do not start until you lyse the cells. And also you should use protease inhibitor as we discussed in the last question. As a general rule, I need to have EDT in a buffer unless it is a problem for specific proteins. So what will EDTA do? EDTA is basically a divalent cation chelator. Okay. So if your protein requires Mg-fed activity, be sure to account for the EDTA in your assay. So what basically these divalent cations sometimes help proteases in their action. So if you chelate the divalent cations by the addition of EDTA, then your proteases will turn inactive. Right? PMSF is also very effective but used usually only during the early steps. Now, and also they said you have to keep everything cold because pre-chill rotors, centrifuge, bottles, tubes, etc. We cannot use anything at room temperature of 25 degree that we have here. Always keep your fractions on ice. When sonicating, do it in short bursts with intervals so to cool the sonication tip and sample in between. Everything should be done in ice and should be kept cold. So as I said, these are the prominent inhibitors. PMSF, it inhibits serine proteases, also inhibits cysteine. If you meant EGTA, then it obviously inhibits proteinases because it basically EDTA, EGTA, they act as divalent cation chelators. So the metalloproteinases, the proteinases which basically needs the divalent cation, their activity is hampered. Then we have peptidine which inhibits aspartic proteases. We have leopeptine which inhibits serine and cysteine proteases and all of that. Now we can commercially buy PIC or protease cocktail mixes which are basically nothing but mixture of all these protease inhibitors in one complete tablet that can stop a multitude of proteases, including serine proteases, cysteine proteases, and metalloproteases. So that is what people use in lab. They buy these commercially and then they use it in their buffers. So bacteria, as I said, lysis for lysis of bacterial cells, you can basically bacteria are typically quite resistant to lysis and special methods are required for breaking open the cells. Cell wall, the cell membranes are pretty resistant. So gram-positive cells tend to be more troublesome than gram-negative cells. So we can do sonication, French press, enzymatic dilation of the cell wall, like using lysozyme and all of that, or we can use repeated freeze-thaw cycles. Basically, all of these are mechanisms to lyse the bacterial cells. Now, the precipitation steps. The solubility of proteins varies according to the ionic strength and hence according to the salt concentration of the solution. So, at low concentrations of salt, the solubility of the protein increases with salt concentration salting in. However, as the salt concentration is increased still further, solubility of the protein begins to decrease. At sufficiently high ionic strength, the protein solubility will have decreased to the point where the protein will be almost completely precipitated from the solution. So in practice, ammonium sulfate is a salt commonly used since it is highly water soluble, relatively cheap and available at high purity. Right? So furthermore, it has no adverse effects upon enzyme activity. Okay. Sometimes we use concentrators as well. Previously, salting out was used to concentrate proteins in addition to the fractionation. And concentration of proteins is now often done using commercially available ultrafiltration devices that concentrate proteins much more quickly and efficiently. Okay. Now, if we talk about the ion exchange chromatography, it's self-explanatory from the name. An ion exchange chromatography resin basically consists of an insoluble matrix with charge groups covalently attached. Now, negatively charged exchanges bind positively charged ions, which are the cations, and they can bind one type of cation, but when presented with the second type of cation, this may displace or exchange with the first. Hence, these resins are called cation exchange resins. Similarly, Anion exchange resins are positively charged and bind negatively charged ions in the anions. Okay. So proteins are nothing but charged molecules, right? So the overall number of charges on a particular protein at a particular pH will depend on the number and type of ionizable amino acid side chains group it contains. For any protein, there will be a pH at which the overall number of negative charges equals the number of positive charges, and so it has no no charge, net charge. This is known as the pI, or the isoelectric point. At this pH, the protein will not bind to any ion exchange resin. Below this pH, the protein will have a net positive charge and will bind to a cation exchanger, whilst above this pH, it will have a net negative charge and will bind to an anion exchanger. Okay, so we have different types of resins like CM resin or carboxy methyl resin is basically a negatively charged cation exchanger. DEAE or diethyl aminoethyl is a positively charged resin, or it is known as an anion exchanger. We have already talked about gel filtration chromatography. I'll keep brief. A wide range of biological molecules can be separated based on difference in size and shape, which lead to differences in their ability to penetrate porous matrices. So, as we discussed in the last question only, this procedure is also known as molecular sieve chromatography. That's the sieve, or molecular extrusion chromatography. It is important to know that shape is a very common characteristic in gel filtration chromatography. Determination of molecular weight by this method is fraught with difficulties and complications. So, combined with other methods such as light scattering, can be very powerful for determination of the molecular weight of proteins and complexes. Only this. So two proteins of very different size can maybe behave similarly on gel filtration column if there is similar radius of gyration. Also, multi-domain proteins connected by flexible linkers can also behave quite atypical. So, as I said, the mechanism, large molecules will not be able to penetrate the pores of the resin and elute in the void volume. Medium sized molecules will penetrate to some degree and elute in a volume between the void volume and the VI. And the small molecules are so small that they penetrate the pore volume of the support and completely elute in the total volume. So, now what is hydrophobic interaction chromatography? So HIC is basically separates proteins with different hydrophobicities based on the reversible interaction of a protein and a hydrophobic surface. So even soluble proteins will have a significant amount of hydrophobic characters, such as 70% of the amino acid or residues on the surface of a protein may be hydrophobic. So high ionic strength stabilizes hydrophobic interactions. Therefore, the samples are loaded in high salt and bound proteins are eluted with decreasing salt concentrations. So ammonium sulfate is often used as the salt. So this is an ideal chromatography method to follow an ammonium sulfate precipitation step. Right. Affinity chromatography discussed for the nickel NTA is basically separates on the basis of a reversible interaction between the proteins and a specific ligand attached to a resin. Typically, it is highly selective and therefore has high resolution and high capacity. Right. So the purification of the specific fusion proteins like histag, as I said, for the nickel NTA chromatography, are all examples of affinity chromatography. So, column generated using antibody specific for the protein of interest is another example of amino affinity chromatography. Okay, so these are all considerations. And what are the how what will be the types of contaminants, that will depend very much on the source of the target protein. For example, fermentation, animal tissue, plant material, its location, all of these will be, these are the typical classes of contaminants found in protein purification, and how to basically avoid these or separate these. Like particulates, like it will include cells and cell debris, usually removed initially using centrifugation or filtration. Then proteins, which will include general host cell protein and proteins of similar properties to the target protein. Then lipids, lipoproteins, polyphenols, nucleic acids that are released during cell lysis and may increase sample viscosity. They are removed using ion exchange, chromatography, and all of that. So similar. So like this include inclusion bodies which are solubilized using chaotropic agents such as 6 molar urea. Aggregate contaminants may be also used here. One sec. One second. Okay. Protein structure will not go into different, but the properties of protein will be determined by the chemical groups which are present, determined by the chemical groups which are exposed on the surface and the overall molecular weight of the molecule, which determines the Stokes radius. Like for example, charge is a very important criteria, measure of the surface ionic properties of a protein giving the molecule a net negative or positive charge. Charge varies with pH, and the isoelectric point is the pH at which the net charge is zero. The presence of sites on a protein which can interact with other biological molecules with a high degree of affinity. Such interactions can be used to purify the target protein. A measure of the water heating character of protein. Highly hydrophobic molecules such as membrane proteins may be insoluble without the addition of the solvents or the detergents. Highly hydrophobic proteins also precipitate at low concentrations of ammonium sulfate. Now, molecular weight is usually measured by gel electrophoresis, the Stokes radius, which gives an indication of shape, solubility and stability, the ability for protein molecule to basically remain in solution is influenced by the pH, salt concentration, presence of solvents and detergents, hydrophobicity, and biological affinity. Okay, so proteins are purified from a wide range of sources, be it blood, animal tissue, plant tissue, fermentation, all of these. Okay, we have talked about the key steps in purification as well. So while no two periods are the same, obviously you have to basically sit down and know the basic properties of the protein that you're trying to purify, and you have to design your purification protocol accordingly. Okay, so the majority of the purifications basically aim to achieve the same results. These are the release of target protein from the starting material, removal of solids to release the protein in the supernatant, removal of water to concentrate the proteins, and removal of contaminants to achieve the desired purity, stabilization of the target protein, and all of that. Okay, so what are the stage 1, as I said, will be the initial fractionation, right? Stage 1 will be the disruption, obviously, of the cell wall, cell membrane. Then the initial fractionation, where the goal is to prepare the protein as a clarified solution. This typically involves technicians to the centrifugation, microfiltration, cell lysis, everything and all of that, right? Then stage 2 is purification, which basically means initial fractionation is followed by a sequence of operations designed to remove contaminants and concentrate the product further, right? Then stage 3 is polishing, the removal of aggregated or degraded protein is also important uh, at this stage, using size exclusion chromatography as a key step, right? Okay. Next we have so spectrophotometer is obviously very important. It's as an analytical instrument in the purification lab. They should also allow measurements of absorbance in the UV and the visible wavelength, all of that. And common uses are in total protein determination and in specific assays such as enzyme kinetics measurements, etc. etc. Right? The second essential item is gel electrophoresis setup. Electrophoresis is the principal method for generating information on a composition of a protein sample, including the approximate molecular weight, isoelectric point of the target protein, and main contaminants. Like, okay, so you have sodium chloride sulfate polyacrylamide gel electrophoresis (SGS page) and isoelectric focusing, which are the two commonly used techniques for determining approximate molecular weight and isoelectric point, respectively. So, in most cases, analysis will involve measurement of the mass of the protein in the sample and quantitation of the specific property of the target molecule to provide values for the yield. Thus, the calculation of specific activity of given fractions to the purification process provides a valuable indication of the level of the purity attained. Okay, so first is total protein quantification. So proteins show maximal ultraviolet absorption at about 280 nanometer, arising from the contribution made by aromatic residues and to some extent from cysteine groups. Absorption at 280 nanometer can therefore be used to provide an approximate protein assay, since the intensity of absorption bears some relationship to the number of these residues present in the protein. All of that, okay. So we'll not go into all of these different assays for this question at least. Yeah, so first is protein extraction of source material, solubilization of proteins before analysis, ideal sample preparation, disrupts all non-covalently bound proteins and removes interfering compounds. Cell disruption lysis, protection from proteolysis, sample fractionation, protein extraction, solubilization, contamination removal, and quantification. So, to facilitate effective disruption of cells or tissues, to isolate proteins from intact cells and tissues while avoiding loss of modification of proteins, to obtain all samples or proteins in sample. It has to basically maximize the sample recovery, retain structural integrity, and all of that. cell disruption, protection from proteolysis, homogenization and sample solubilization, right? So general disruption is employed when sample consists of easily lyse cells like tissue culture cells, blood cells. They're employed when particular subcellular fraction is to be analyzed, like gentle osmotic suspense cells in hypotonic medium, uh, suspense cells in detergent solution, cellulase plant cells, uh, lysozyme bacteria, rapid freeze cycles for this. So cell lysis vigorous disruption will be employed when sample consists of cells which can't be easily lysed, like bacterial and plant cells. Harsh lysis like sonication, French pressure, homogenization, and all of that. Then manual grinding, all of that will be vigorous. And overview lysis method. So these are the lysis method: tissue culture cells, gentle, detergent, enzymatic lysis, freeze thaw process, French pressure, gas breed, grinding, mechanical homogenization, osmotic lysis, sonication, etc. etc. So this is protection from proteolysis is an important part. Basically, cell lysis will also release proteases, which may result into proteolysis. During the sample preparation, protease inhibitors are added to minimize the artifactual proteolysis, right? So effective protease inhibitors contains a mixture of irreversible and reversible protease inhibitors that inhibit serine, cysteine, and metalloproteinases, specifically useful for gel-based proteomics. Like PMSF is phenyl methyl sulfonyl chloride, effective against serine and cysteine proteases, inactivated by DDT, unstable. So ethylene diamine tetracetic acid, effective against proteases, inhibits nucleases as well. And that EGTA basically effective against metalloproteases. Self-processing in cold conditions also reduces proteolysis. So why fractionation? Because it makes it possible to isolate groups of proteins or fractions from total proteome. So it basically simplifies the analysis of complex protein mixtures, allows for improved resolution when an individual fraction is analyzed, and provides less crowded 2D maps. Okay. So it can be centrifugation like subcellular fractionation organelles, mitochondria, cell compartments like cell membrane. Then we have methods like abundant protein removal like affinity chromatography. Then we have sequential extraction based on solubility and electrophoresis, liquid phase, IEF, fractionation and all of that. So, importance of fractionation increases proteome coverage, separates highly abundant proteins from low abundant proteins of interest, and bring them into detergent dynamic detection range. So, it increases chances of identifying low abundance proteins of diagnostic or therapeutic interest. Now, protein extraction and solubilization. Now, protein extraction preceded by subcellular fractionation to enrich proteins of interest. Protein extraction in aqueous buffer, like trace hydrochloric acid followed by desalting. Protein precipitation by trichloroacetic acid, acetone, TC and acetone. So protein extract should be soluble, free from protein, DNA, RNA, protein interactions, metabolites, etc. Now, sample solubilization proteins naturally form complexes with membranes, nucleic acids, or other proteins. Form various non-specific aggregates. Precipitate when removed from their normal environment. Right? Now, effectiveness of solubilization depends on choice of cell disruption, protein concentration and dissolution method, choice of detergent, sample composition, etc. Okay. Now, solubilization methods differ from sample to sample. Now, urea is used as a denaturant to solubilize and unfold most proteins to fully random conformation. Urea is a chaotropic agent, helps in stabilization and folding of proteins. All ionizable groups exposed to yeah. So high area like improve solubilization of membrane proteins and all of that. Detergents like extremely efficient in solubilizing hydrophobic proteins. However, anionic nature limits its effectiveness for conventional proteomic analysis. This is not compatible with isoelectric focusing as well. So basically, ionic and non-ionic detergents are used for proteomic interaction studies. So DTT solubilizing agents we have already discussed. So basically different types of biological samples, different types of challenges. For example, fungal cells have tough cell wall proteolysis problem, tissue culture cells and interference from salt serum. So depending on your source, you have to optimize the protocol. Right. Right. We'll not go into serum proteomics or anything. We have talked about all of this chromatography. We have talked about page as well. So coming back to the question that we initially started with, the correct order of event in the general strategy of protein purification from tissue of microorganism is this. First is disruption, then crude fractionation, then selective fractionation. So if you have any questions, you can ask me now, or we can go to the next question for today. I'll give you a few minutes and we can go to the next question for today. Okay. Then to the next question for today. With the known knowledge of the protein you want to purify from a natural source, a size exclusion chromatography experiment provided several proteins, and for the characterization of the extracted protein, both molecular weight determination and secondary structure analysis are required. What will be the proper approach of determining exact molecular weight and secondary structure? Anybody? Okay, so the correct answer will be MALDI, followed by CD spectroscopy. So a little introduction into MALDI. So, MALDI is assisted laser desorption ionization time of flight mass spectrometry used to detect and characterize mixtures of organic molecules. In micro, used as a rapid, accurate, and more cost-effective method for identification of microbes, right? So mass spectrometry basically measures mass to charge ratio, right? M by C of ions. The results presented as a mass spectrum or plot of the ion signal as a function of the mass to charge ratio. And spectra is used to determine spectra, the elemental or the isotopic signature, and the masses of particles, the chemical ID or structure. Now an ion source sample is ionized for solids by electrons of MALDI. Ions then separated based on mass to charge ratio done by acceleration and subjecting to electric or magnetic field. Ions with a lower mass will reach the detector first. A detector records either charge or current produced when ion passes by or hits the surface, produces a mass spectrum, usually an electron multiplier, right? So multi-TOF MS will have an ion source, laser and ionization chamber to ionize sample and transfer into a gas phase. Uses laser energy absorbing matrix to create ions from large molecules. It will have a mass analyzer, separates ionized analytes according to their mass, all same charge. TOF will use electric field to accelerate ions and measure time to reach detector. A detection device to monitor separated ions. Okay, so procedure will be combined acylated colony, formic acid and matrix will multiplate. So matrix isolates molecules from each other, protecting them from fragmentation and enabling desorption by laser energy. Now consists of small crystallized acid molecules, usually synapenic acid, alpha cyano and all of that. Now ion sources laser uses UV lasers. Laser pulses fired at the matrix crystals in the bright Doppler spot. The matrix absorbs the laser energy, converting into an ionized state. Charge is transferred to analyte. Random collision in the gas phase. Ionized analyte and matrix molecules are dissolved from the plate. Laser beam desorption, desorption or ionization and return transfer to mass analyzer. Now, mass analyzer is analyzed hydrogen molecules accelerated through a positively charged electrostatic field into a time of flight tube. Inside vacuum tube, ions travel toward an ion detector. Small analytes travel the fastest. Ions emerge from the mass analyzer and hit the ion detector, generate a mass spectrum representing the number of ions of a given mass impacting the detectors over time. Okay, like this. If you see, the laser is going to sample goes to detector, and all of these. So mass spectrum provides profile unique to individual types of microbes with peaks specific to genera and species once acquired compared to database of reference spectrum or value percentage score is generated. This is how it is done. Higher the log or score, higher the similarity between the mass spectrum and the of the isolate and the database entry in the reference. So advantages is rapid, inexpensive, because no reagent cost, small amounts of organism are required, direct sample entry possible, reduced labor. Limitation, databases are proprietary unlike publicly available signal databases. Difficulties in identification with some organisms, difficulty analyzing mixed cultures, identifying microorganisms with liquid cultures, low identification scores, human error, all of that. Okay. Now, what is circular dichroism or CD? CD is basically its spectroscopy method measures differences in the absorption of left-handed polarized light versus right-handed polarized light, which arises due to structural asymmetry. The absence of regular structure results in a zero CD intensity, while an ordered structure results in a spectrum which can contain both positive and negative signals. Right? Chiral structure can be distinguished and characterized by polarized light. Optical rotation, the rotation of linearly polarized light by the sample. Optical rotary dispersion, the variation of optical rotation is a function of wavelength. The spectrum of optical rotation. Circular dichroism, the difference in absorption of left and right circular light. Now, plane polarized light consists two circularly polarized components of equal intensity. Two circularly polarized components are like left and right-handed springs. So, as observed by looking at the source, right-handed circularly polarized light rotates the clockwise. Frequency of rotation is related to the frequency of the light. Can be resolved into two circularly polarized components. So, when added together after passing through an optically isotropic medium, plane polarized light resolves. Okay, so passing plane polarized light through a brief birefringent plate which splits the light into two plane polarized beams oscillating along the different axes. When one of the beams is retarded by 90 degree, then the two beams which are now 90 degree out of the phase are added together and the result is circularly polarized light of one direction. So by inverting the two axes such that the alternate beam is retarded, then circularly polarized light of the other direction is generated. The result of adding the right and the left circularly polarized light that passes through the optically active sample is electrically polarized light. So the circular dichroism is equivalent to electricity. Okay. So we will not go into very much detail. So basically measurement of how an optically active compound absorbs right and left-handed circularly polarized light. That is all the time. So difference between the absorption of left and right-handed circularly polarized light, and it's measured as a function of wavelength. CD is measured as a quantity, mean residue electricity whose units are degrees centimeter square per d mole decimole. We're not going to do much detail. So coming back to the question that we started with. Yes, the question that started with the knowledge of the protein you want to purify from a natural source. So, a dissection chromatography experiment provided several proteins for the characterization of the extracted protein. Both molecular weight determination and secondary structure analysis are required. So, it will obviously be MALDI for the exact molecular weight, followed by CD for secondary structure determination. Anybody has any questions related to queries related to this question? I'll give you 2-3 minutes. You can ask me now. Okay, so if you have no questions, then we can stop the session for today. We'll meet again next Monday, same time. Thank you. Thank you. You can leave the session.